here to give a talk. So it's my great pleasure today to have Dr. Lei Zhang here to give a talk in our immunological seminar series. So Lei got her bachelor's degree from Minzu University of China in 2009, and then obtained her PhD degree from Peking University in 2016. After that, she joined Zemin Zhang's laboratory as one of the earliest lab members for postdoc training. During this period, they focused on understanding the immunotherapy mechanisms in colorectal cancer on the single cell level. In collaboration with her colleagues, she established a fantastic single cell transcriptomic analysis platform to elucidate how different tumor infiltrating T cells and myeloid cells functions during the immunotherapy, such as anti-PD-1 therapy and anti-CD40 treatment. With this outstanding works, in early this year, she got her faculty position and started her own lab at Shenzhen Bay Laboratory. She's also a joint faculty of Peking University Shenzhen Graduate School. So now, without further wasting the time, I will pass the microphone to Dr. Lei Zhang. So please.
Thank you more for the nice introduction. I'm Lei Zhang from Shenzhen Bay Laboratory and Peking University Shenzhen Graduate School. I'm glad to share my recent work. My talk is about dissecting the mechanisms of immunotherapy by single-cell analysis. As we have known that cancer is a severe disease to threaten human health. Here are 10 hallmarks of cancer. 2 of them are avoiding immune destruction and genome instability and mutation. Regarding to immune destruction, researchers study focused on T cells and their immune checkpoints have greatly enhanced the development of immune checkpoint blockades, such as PD-1 and CTLA-4 blockades. And such studies have enhanced the such strategies have dramatically altered the clinical treatment. We have observed favorable treatment response in melanoma and lung cancer. However, the treatment responses to immune checkpoint blockades are not uniform in different cancer patients. For example, for colorectal cancer patients, macrosatellite unstable patients showed significantly better treatment responses to PD-1 blockade than macrosatellite stable patients. However, the underlying mechanisms were not clear, so it's quite needed to understand what the fundamental properties of T cells in CRC and what are the differences of T cells between these 2 groups of patients. As we all know that the tumor microenvironment is highly heterogeneous and complex. Beyond T cells, there are several other immune infiltrating cells, such as myeloid cells. Besides T-cell-based immunotherapy, such as PD-1 blockade, recent studies have been focused on the myeloid-based immunotherapies. However, the mechanisms of these therapies has not been demonstrated clearly enough. So understanding the complexity of the tumor microenvironment is quite critical for identifying immune modifiers for effective treatments. But it's difficult to study the tumor microenvironment by traditional methods. My study focused on using single-cell technology. Here I will give a brief introduction for single-cell technology. This technology was initially applied to transcriptomes named single-cell RNA sequencing. It was first developed by Fu Choutang from Biopic PKU when he was a postdoc in Sanger Institute. In that work, they studied the whole transcriptome from a mouse embryonic cell. After that, different single-cell RNA sequencing methods has been developed. Two of them full-length sequencing named SMART-seq and SMART-seq2. More recently, the droplet-based methods has been commercialized, as we all familiar with its name, 10x genomics. Beyond transcriptomes, single-cell technologies has been applied to study different layers of a cell. For example, single-cell ATAC was developed to study the epigenomic alterations. For genomic information, although it's difficult to obtain, we still have several single-cell DNA sequencing methods to obtain the genomic alterations, such as copy number variation, structural variation, and even recently we can obtain the new mutations. And more recently, single-cell technology has been integrated with other technologies such as imaging, mass spec, tetramer, and these methods has been the experimental and the analytical methods for this integrated technology has been being developed. To enable us to obtain the spatial transcriptome, epitope, and perturbation of a cell. Since as this technology matured, we can obtain this multi-omics from a single cell. My study started from about 5 years ago near this period. So we use single cell RNA sequencing. Here are the major experimental procedures for single cell RNA sequencing. We can obtain single cells by either FACS or MAX, as we have familiar with. The most difficult step for this procedure is the amplification. It can be divided to two major methods. For the plate-based methods, we can use the pooled PCR amplification, such as the droplet-seq, cell-seq, and MAR-seq. For individual cell amplification, we can use the SMART-seq and SMART-seq2 methods. These methods can obtain, as we have mentioned, can obtain the full length of the transcriptome, which enable us to obtain the whole genome and more transcripts. But is relatively high cost. By contrast, the pooled PCR amplification can only obtain 3'. More recently, can obtain the 5' of a gene of a transcript. So the detected gene would be lower, but the throughput of a cell could be expanded. So all of this technology could have advantage and limitations, we choose the full length sequencing SMART-seq2 as a beginning method, and more recently combined with the droplet based methods. Single-cell technology was initially used to study the embryonic development. For the study of tumor microenvironment, here are two remarkable publications. One from Aviv's lab. They published the single-cell analysis of 10 immune and non-immune cells from 19 melanoma patients. This paper has been published on Science in 2016. In the same period, Bermin is pioneering in single-cell analysis of T cells in cancer. Zhang's lab published the first paper on Cell in 2017 about the single-cell analysis of T cells in liver cancer. As this field advanced, several key questions are raised, including what are the fundamental properties of different immune cells in the tumor microenvironment, and beyond such profilings, on how to trace the dynamic relationships of T cells and other immune cells, and how to use single-cell analysis to dissect the mechanism of immunotherapies. So to answer this question, I conducted my postdoctoral projects mainly focused on the tumor single-cell analysis of the tumor microenvironment of colorectal cancer. My first project is linear tracking of the dynamic relationships of T cells and published about 3 years ago. And my recent work is about the myeloid studies in CRC. So in the next section, I will give a brief introduction of the key findings from the T cell study and make a focused discussion on the myeloid study. In the T cell study, we have collected T cells from tumor, normal adjacent tissues, and the blood from 12 colorectal cancer patients. And after a series of experiments, we obtained over 10,000 single full-length transcriptomes from single T cells. And we obtained 20 T cells from colorectal cancer with distinct gene expression patterns and different tissue enrichment pattern, such as CD8 exalted T cells, Th17 cells, and Tregs were preferentially enriched in the tumor of colorectal cancer patients. For the lineage tracking is quite different. It's quite difficult to track the dynamic changes in human tissues. So to over these difficulties, I have collaborated with my colleagues to use the TCR as tags to develop a novel analysis framework named StatTrack to quantitatively evaluate the clonal expansion, tissue migration, and the state transition of T cells we identified from CRC patients. After developing this framework, we asked what novel biological findings can we obtain. One of the example is related to the mechanism of MSI patients. We identified among several CD8 and CD4 T cells which could produce interferon gamma. We identified that a T helper cell subset named Th1-like cell was peripheral significantly enriched in microsatellite unstable patients. you may remember that these patients showed favorable treatment responses to PD-1 therapy in clinic. This T helper cell different from traditional Th1-like cells because it has low expression of TBX21, which encodes the transcription factor Tbet. But it used its interferon gamma expression was governed by another transcription factor named BHLHE40. By Star Trek analysis, we identified the clonality of Th1-like cells and identified that these cells were preferentially were significantly clonally expanded in MSI CRC patients and have the most higher proliferation score in the microenvironment. By further in vitro analysis, we identified that its cell surface protein IGFLR1 and its ligand IGFL3 could be a potential co-stimulatory pathway for CD4 cells. So all of these data suggest that the Th1-like cells could have anti-tumor activity. So based on this study, we asked, can we boost anti-tumor immunity of Th1-like cells in colorectal cancer patients, especially for microsatellite stable patients who cannot benefit from current immunotherapies? And can we use other immunotherapeutic strategies such as myeloid-based immunotherapies? So my next study was based on these questions. To understand the fundamental properties of myeloid cells in CRC, we also conducted the single cell analysis. The experimental pipeline was similar to T cells, to my previous T cell study. The difference is that as the technology developed, as we mentioned, we used two RNA-seq, single-cell RNA-seq platforms and combine the advantage of these two methods. We obtained different myeloid groups with distinct tissue-enriched pattern. For example, we identified 3 groups of myeloid cells enriched in the tumor of CRC. One group highly expressed FCN1 named monocyte-like cells, and 2 groups of macrophages enriched in tumor, which defined as tumor-associated macrophages, that are TAMs. Since we have obtained we have characterized the T cells in my first study and myeloid cells in this study. So we asked, what are the relationships between these immune and non-immune cells in CRC? To answer this question, we performed computational modeling of single-cell RNA-seq and from our study and bulk RNA-seq data from TCGA CRC patients. We finally constructed this interaction network in CRC. As we can see that 2 groups of deceased and 2 groups of TAMs are in the core of this interaction. So we then asked whether and how DC-based immunotherapy and TAM-based immunotherapy could alter this interaction and influence their anti-tumor immunity. For the DC-based therapy, we used the CD40 agonist. Since it were CD40 treatment has showed showed effective treatment response in preclinical study and in about 6 clinical study. But how CD40 altered the tumor microenvironment was not clearly enough. So we conducted single-cell RNA sequencing from mouse tumor models treated with anti-CD40 agonist. At first, we compared the myeloid subsites from mouse tumor models and the human CRC patients. We observed conserved pattern of these different DC groups from these two species. As we can see that we identified two groups of CDC1 cells. CDC1 cells. One is immature CDC1 cells, high expression with collectin 9A. The other characterized as high expression of CCR7 and CD40, named activity CDC1 cells. Interestingly, this DC group were resembled with LAMP3 DC identified also in Zhang's lab in liver cancer, and show similar patterns across different cancer patients, including lung cancer and UC patients and mouse models. So we asked how the CD40 treatment altered these different DC and TAM populations. We identified that after CD40 treatment, the CCL22 positive CDC1 cells were significantly elevated by such treatment. And it was also activated by CD40 treatment as marked by a high level of CD80 and CD86 and elevated expression production of IL-12. So we know that IL-12 is important for DC to prime T cells. So we asked what the downstream changes of T cells. For CD8 T cells, we observed that after CD40 treatment, CD40 agonist can significantly increase the proportion of CD8TM and CD8TRM, but some sort of reduced the proportion of CD8 exalted T cells. For CD4 T cells, there are several groups of CD4 T cells highly express the CD4's ligand CD40LG. Among this CD4 T cells, we observe that Th1-like BHLH40 positive Th1-like cells were significantly elevated by CD40 treatment. And by StatTrack analysis we developed in our T cell paper, we also observed that the Th1-like cells were the clonal expansion was also enhanced by CD40 treatment. Since we observed the alterations of activity DC groups and the T cell groups, we asked what the relationships between these two groups of immune cells. We performed TCG data analysis and the in vitro experiments. And by the TCG data analysis, we observed the direct correlation between activated CDC1 cells and the Th1-like cells in this purple bar, and we can see that these two groups were enriched in MS high MSI colorectal cancer patients. Take a summary of this part. By using the combined single-cell analysis, we identify the potential mechanism of anti-CD40 treatment with alterations of CTC1 cells, especially Th1-like cells and different CD8 T cells. As we previously introduced, that there are also two groups of TAMs in the core of the interaction in CRC. As we can see in this figure, that C1QC positive TAMs showed preferential interaction with different groups of T cells, while the SPP1 positive TAMs showed interaction with cancer-associated fibroblast and endothelial cells. This inferred interaction suggests that this C1QC TAMs and SPP1 TAMs could have different function and phenotype in the tumor microenvironment of CRC. To address this hypothesis, we performed additional analysis. Firstly, we identified that the traditional M1, M2 phenotype could not explain the differences between C1QC and the SPP1 positive TAMs. But by further downstream volcano analysis and pathway analysis, we identified that the C1QC positive TAMs showed pathways towards complement activation and antigen presentation, antigen processing and presentation, while SPP1 TAM showed positive TAM showed pathway towards tumor angiogenesis, even the colorectal liver metastasis. By lineage tracing analysis, we identified that both C1QC and the SPP1 TAMs were derived from from circulating monocytes. This was in line with the observation from mouse models, but in detail, this C1QC and SPP1 TAMs showed different origins from tissue resident macrophages. So we asked what are the effects of manipulating TAM populations? To answer this question, we used the CSF1R blockade. This treatment was supposed to be deplete macrophage in the tumor microenvironment to shrink the tumor size. However, the treatment responses of this therapy were limited in clinic, but the underlying mechanism was not clear. So we also used our expertise in single-cell technology to obtain the sequencing the single-cell data from mouse model treated with CS1R blockade. Similar to the DC-based therapy study, we also carry out the comparison between macrophage from mouse and human. Different from the highly conserved pattern of DC, macrophage showed some conserve and in conserve pattern, but we still identified macrophage population in the mouse model showing similar similarity with those from human CRC, such as the SPP1 positive TAMs. After treatment, however, we observed that CSF1R almost depleted two of macrophage populations, but one population were significantly remained even elevated. Further analysis identified that the depleted macrophages population showed signature towards inflammation and high and this population and hair proliferation potential, but the resistant CSF1R resistant TAM population preferentially expressed high genes involved in angiogenesis, such as VEGF-A and immunosuppression. So take a summary of this section. By using the single-cell analysis, we identified the treatment-resistant mechanism of CS1R blockade. More importantly, we point out a population named SPP1 positive TAM and several signature genes that could be serve as potential therapeutic targets for next generation immunotherapy. Take a summary of my talk. By using single cell analysis, combined single cell analysis from colorectal cancer patients and mouse tumor models with treatments, we established the interactions of myeloid cells and with different immune and non-immune cells, and identify the mechanisms of two myeloid targeted immunotherapies, including DC-based immunotherapy, that is CD40 agonist, and TAM-based immunotherapy, that is the treatment-resistant mechanism of CSF1R blockade. As the last, I would like to give my special thanks to my postdoc supervisor, Professor Lei Zhang, and the T-cell study were collaborated with our team member, Liang Tao and Yuan Yuan. The myeloid study were collaborated with Dr. Zi Li and Fang Qiao. Our collaboration from the industry is Dr. Wenjun Ouyang from Amgen and his team member, including Xin Jackson and Kasia. The human CRC colorectal cancer sample were obtained from Peking University Renmin Hospital. As Mo has introduced, I have moved to Shenzhen this year and started my own lab at Shenzhen Bay Lab and Peking University Shenzhen Graduate School. Now I have recruited one postdoc. His name is Jason Lee. He will carry out our single cell analysis in the future, and we have 3 RNAs in our lab. We also have open positions for assistant PIs, postdocs, and PhD students. So I will very appreciate for your recommendation. So thank you for your time, and I'm glad to answer your question. Thank you. Hello.
Hey, Lei. This is really a powerful tool. Very inspiring. I think I might have a technical question, and hope you can help me to understand. So the lineage tracing was achieved by like applying these isotope at different time points, right?
You mean my lineage tracing in the first study names?
More about in the second, like the CD41.
Oh, yeah. The tracing for lineage is also used the tool we developed in CRC T cell study. So since because we can capture the TCRs from the single cell data and the transcriptomes, so we can use both the information. Actually, we use the clonal tab. If we have, maybe I can make an example. If we observe the clonal tab, we can we can see maybe 40 cells. It can 10 cells come from such as exhausted T cell, and the others come from um, effector memory T cells. So we can imply they have the transition. If the clone tab comprised from different T cells from different tissues, maybe like from blood or tumor, so we can imply the tissue migration of these T cells.
Okay, I see.
For yeah, for this is for the T cell study. For the myeloid study, because we don't have TCR,
Mhm.
something like TCRs in the myeloid study, we only have transcriptomes. So we use the different how to say inferred developmental trajectory. We use different methods. One method is based on their similarity, transcriptome similarity. If one cell show um, transcriptome similarity to the other cells, so we can line them one by one. Another method is using their RNA velocity. If I mean, so if the unspliced and the spliced information could have the time point, so we can infer their trajectory. That's for the myeloid study.
Okay, and because I remember you have one of these, and so these are more about maybe tracing the effect of the so when you like at different time point you give the isotope and then
Oh, you
you
mean
collect the
yeah.
sample in the end. So are these like you're giving the same like so are these like isotope of the like amino acid or like the building blocks for the cell survival, and are you giving always the same one or it's all a different one, different time point so that you can infer their like their development in the end?
Yeah, that's a very good question. Actually, we gave the same amount of treatment for the CD40 treatment. We have different point.
Oh,
Uh, yeah, we gave the treatment on day 2 and day 10, and so we can observe the alterations. But we don't know whether the treatments have influenced their proliferation or something have long-term effects. Yeah.
mhm, mhm. I see. The other maybe a practical question, which is, let's say if you get a tumor, which is like a big chunk, uh, does it matter like which part of, are you like, can you take all the cells in that tumor or due to the limit of the handle capacity, usually we can only take a small piece of tissues. But then the question is which piece to take from. Does it matter? Like I think it might like matter if it's from the, let's say, the center versus the peripheral of the tumors. Like if there's any like variation came from the position of the
Yeah, that's very sorry. That's very important, especially for the sampling. I think you are curious. You are mentioning about the sampling precision. Yes, for tumor cells, is well known. Is also already known that the heterogeneity of the precision for the immune cells. For the immune cells, we becoming to know that there would be different, there would be influence for the precision on this immune microenvironment, even in the same tumor. So at the beginning of my study, to avoid such precision influence, we obtained the single cell from a piece of tissue from a whole resected surgical resected tumor. But to avoid such influence, we try to capture the same precision from different cancer patients. But right now, I remember for therapy studies, they will use the biopsy. Since it's still a very important question, so recently different groups using the spatial transcriptome to study this question, whether there would be position or structure influence in the tumor microenvironment, especially to immune cells.
I see. So yeah, since you mentioned the spatial transcriptome, so is that like inferred from the single cell like gene expression profile, or there is a like you can physically look at which cell is next to which?
There are several different methods. Some combined with the imaging methods, so you can using the dissection from the tissue, so you can gain the information directly, and the other methods combined with single cell transcriptome, so you can perform IHC at the same time and infer. Yes, you will use the infer information from single cell.
I see. So let's say
I think it's still being developed, especially including experimental for the imaging methods, for the imaging-based methods. Although it's accurate, but the throughput is limited because you can only label hundreds to 1,000 genes, but it is still limited. But the for single cell spatial transcriptome, the analytical methods need to be developed. Yeah.
I think there's these like you can laser cut like the cells after
yeah.
But this probably is like also technically challenging. I mean, ideally it will be because there's some maybe bioinformatic way that you can like also infer the spatial organization from based on let's say the expression of the receptor and the ligand pair on different cells. I think that probably will involve like some bioinformatic work in the bio. Will be interesting to do. Okay, good. Yeah, thanks so much.
Thanks, Xiaolei.
Hello. Can I ask questions?
Yeah.
Hello. Okay, right. Thank you very much for hi, this is Dee. Thank you very much for your talk. Very nice talk. I have some technical question. So you have done both the SmartSeq and 10x genomics. And for the clonality analysis, do you see have you used the 10x genomics for clonality study for T cells? Did you see those?
Yeah, it seems the connection is not good, but I think your question is about the clonality from 10x data. Yeah. And the method
between 10x, sorry, yeah, my connection was good at home. Yeah. Anyway, my question is about the difference for clonality analysis for T cells between N times genomics and SmartSeq.
Yeah. Our method named StratTrack was initially developed based on SmartSeq data. Yeah, because it has full length, so you can obtain more information. But in the second study, I mean, in the myeloid study, we also capture T cells by 10x genomics. So we apply the StratTrack analysis in such data. The TCR information from 10x obviously was not how to say the full length sequencing of TCR alpha beta pairs. The frequency of cells you can capture from 10x data is lower than SmartSeq2. For SmartSeq2, we can obtain almost 80 to 90 cells. You can obtain the full length, you can obtain alpha beta pairs. But for 10x genomics, you can, I remember the near 7% cells. So
only 7% cells, because a lot of drop off rates of the
you need to be cautious for TCR alpha and beta pairs. So, but the 10x data is the amount is large. So although 70%, that would be enough for your analysis. Yeah.
Okay. Yeah. The other two questions. The second part of your story, you demonstrate how the myeloid cell interaction with T cells. Can you elaborate a little bit more how you determine this cell-cell interaction?
Maybe I can share my screen again. Yeah. Yeah, this was done by such analysis. If you have performed single cell analysis, especially for interactions, mostly used methods are based on ligand and receptors developed by Sarah Techman from Sanger Institute named cell phone DB. But in our study, we want to use beyond ligand receptor. So we combine the single cell RNA-seq data and TCG data sites. We we obtain these signatures and combine to TCG data, so we can identify which gene were correlated to the abundance. You mean to different groups of immune cells. So after identify these genes, we go back to the single cell data to identify this gene belongs to which group. For example, you can infer the SPP1 positive TAM signature, captures SPP1 positive signature, and then calculate in the TCG data site. Identify maybe genes from CAPS correlated with SPP1 positive TAM. A lot of genes, maybe include CAPS or other genes. So we obtain these genes and go back into single cell data to check these genes belong to which tab. Yeah, that's the major procedure.
Okay, so basically you through the gene association to infer to the cell interaction.
Cell level, yeah.
Okay, so it's from bulk transcriptomics to identify the gene associations, then going to the single cell to look into. Okay, all right, that's a very interesting idea. The other thing I think may not be a very fair question to ask you, but if you have any insight, I would love to hear. From first part of your talk, you were able to identify these CXCL13 and Th1-like cells.
Yeah.
And there's a two question related to that is CXCL13 has been known as a chemokine organized lympho architecture, particular B cell follicle. So in your study, did you see those are more B cell infiltration for those samples with CXCL13 signature? And the other related question is CXCL13 has been known also enrichedly produced by TFH cells. So what's the difference between TFH or this CXCL13 and Th1-like cells?
Yeah, I thank you for question. That's very interesting and was asked by several reviewers when we published that paper. Yeah, I would like to answer the second question. Yeah, because you named some sort of new subtypes, so we didn't mean novel or new in our paper. We just mentioned a subset. So I would like to answer the second question. The Th1 cells, 13 positive Th1-like cells were different from TFH, although they have high expression of PD1 CXCL13, but the CR5 and BCL6 level in these subgroups were very, very low. So we identified TFH cell in CRC, especially in the normal tissue. So we confidently say that they were different groups, but they did share some similarity. Yeah. For the interaction with B cells, we try to answer this question through single cell data, as you have mentioned, that's the interaction analysis. But the B cell from our data was not very large because we aim to answer T cell and myeloid-based therapy, but based questions. So that's very interesting, especially if is there any structure between TFH, Th1-like cell, B cell, and other immune cells. So I would like to answer these questions in my future study. I mean, yeah.
Thank you.
Thank you.
So the next question from Liang Qu. I guess there's a question in regard to microbiota. And Liang asked, did you collect gut microbiota data? Does gut microbiota play a role in the interaction between immune cells and the colorectal cancer?
Yeah, that's also very interesting and important question, since microbiota, yeah, has important role in not only in the interaction but also in immunotherapy responses. But we didn't collect gut microbiota, and also I'm not very familiar with this field. But it's very interesting. If you want to answer such a question, maybe we can collaborate with your expertise, yeah, in the future.
So any more questions?
Yes. Do we have time for one more question? Hello?
So, please.
Hello? Hello?
Yeah, so this is Ganzo from Georgia. It's very powerful technology to address the fundamental question. So I understand you focused on T cells and myeloid cells.
Yeah.
Did you also have the chance to look at the tumor cells, the cancer cells, right? You know, especially the cancer cells from responder versus non-responder to immunotherapy, to see what the signaling pathways are affected by the therapy. And so that may, you know, infer, you know, if it is a successful immunotherapy, you know, how cancer will respond.
Yeah, that's also very important, because we at first focused on T cell and the immune cells, but actually we did collected non-immune cells, including at the beginning, we don't know which population are tumor and which population are non-tumor. So we performed analysis, including inverse CNV, so we can define the tumor cells according to their genomic alterations. Although single-cell RNA-seq is not the best tool to answer the tumor-related question we tried, so we did identify the different tumor populations. I remember there are derived from goblet and epicellular cells, but because the number of these cells were limited, so I tried to combine macrosatellite stable patients and unstable patients to combine the tumor, the differences between tumor cells. I remember the goblet cell were different, but I cannot make the conclusion since the patient number and cell number is limited. But if I can have, recently I am talking with some guy who good at next generation of single cell technology. So if this works, I will take more attention to tumor cells, but right now focused on the immune cells in the tumor microenvironment. Thank you.
Yeah, that's fair. Thank you.
Hi, I have a question.
Oh, okay.
So as time is running out, so let's ask the last question. It's from Dave. So the question is, the SMART-SIG2 data can, if the SMART-SIG2 data can represent tumor microenvironment composition, what could be the explanation for high C1q cell patients with low T cell in tumor microenvironment? What? So the last question.
Cell patients. SMART-SIG2 data. The low, actually, the C1QC, high C1QC patients.
Yeah. In my understanding, the C1QC macrophages is a pro-inflammatory subgroup of the macrophages, yeah. And I'm very interested in your CRC paper because I observed the C1QC subgroup have very high chemokine expression, such as the CXL9, 10, 11. Yeah. Suppose those chemokines will recruit a lot of T cell, especially the CD8, CD4 subgroup T cells into the TME. So but in some patients, the T cell infiltration is very low. So what could be the explanation of the patient have a higher portion of C1QC subset, but have very low T cell in the TME?
Yeah, that's very interesting. Actually, we did perform interaction between TAMs, different TAMs and T cells. As you have mentioned that C1QC produce different chemokines and cytokines. In line with this, we observed the C1QC positive TAMs showed multiple interaction with different T cells, including CD8 T cells. I remember Xu has also observed this patterns phenomena. Yeah, C1QC positive TAMs could interact with CD8 T cells and CD4 T cells. But so I try to compare the TAM population between MSI and MSS because these two patients have different treatment responses to PD-1.